Okay, so welcome to the second video in the bacteriology series, and today we're going to discuss cholera. I was going to do tuberculosis, but I'm not quite feeling up to tuberculosis tonight, so we'll do cholera instead. So let's begin. And it's not really in a great position. I have this problem every single time. The ideal position for the camera isn't the ideal position for me writing. Okay, so bring this down here, never mind. Okay, so cholera. Cholera. So it is a diarrhea disease, and it's caused by the bacterium Vibrio cholerae. Vibrio cholerae. So that's the fancy name for the bacterium which is going to cause the disease cholera. So Vibrio cholerae is a gram-negative bacterium. Gram-negative, which means that it stains red under gram staining, and it kind of looks like this. It's sort of a rod shape, and then it has a long waggly tail that comes like this. So a long flagella, that's the fancy name for this tail. It's a flagella, which helps it to propel itself through fluid, and then inside you have the bacterium. So it will appear red under gram staining. So this is a picture of a Vibrio cholerae. Vibrio cholerae. And you've probably been taught in either history class or basic science class that cholera is waterborne. There's the very famous outbreak in London where it was coming from one well, I believe, or no, I think it was past the stage of wells. I think it was like a water pump, one water pump. And John Snow was the person who sort of evaluated, plotted on the London map all the cases of cholera, and realised that it was focused around this one water pump, and therefore closed the water pump, and the epidemic and the little outbreak stopped. So it's waterborne. So in contaminated water. So if someone who has cholera, if their feces ends up in the water, and then someone drinks that water, they're going to get cholera. So in the water you have Vibrio cholerae. So the Vibrio cholerae is within the water. Then if someone is going to drink this water, so it's going to go down. Well, first it's going to go into the oral cavity, then it's going to go down the oropharynx, into the laryngopharynx, then down the esophagus, into the stomach, and then it will go through the pyloric sphincter into the duodenum, through the duodenum to the jejunum, and then into the ileum. So it colonizes the small intestine rather than the large intestine. So all three sections of the small intestine are going to have this bug in them. And basically it's going to colonize the mucosal surface of the small intestine, and I thought now we can do a bit of revision of the histological structure of the small intestine. So it looks kind of like this. Let me just make sure it all's going to fit. Okay, I need to move it up a bit. So that's a circular cross section, and the outer layer you have is a layer called the serosa, which is basically made up of mesoderm. It's called mesothelium rather than endothelium or epithelium, and it's because it's derived from mesoderm. So most epithelia are derived usually from so the skin, for instance, the epidermis is usually derived from ectoderm, from the embryonic trilaminar embryonic disc, and the epithelium within the gut is derived from endoderm, and then endothelium is the epithelial cells that line the blood vessels. But this is different. This is mesothelium, and it's derived from the lateral plate mesoderm. So when the embryo sort of folds over, and it folds laterally and it folds cephalocaudally, it forms an intraembryonic coelom, which is lined by mesoderm, and this here is part of that old mesoderm that was lining that cavity, because what's happened is that the intestine pushes its way pushes into this cavity and gets covered by the layer of the cells that surround it, and that's what actually makes the mesentery, and that's the peritoneal cavity is part of the remnants of this intraembryonic coelom, along with the pleural cavity and the pericardial cavity. Okay, so that's what this is, that's what the serosa is. So the serosa will be continuous with the layer of mesentery behind. So that's the serosa. And then the next layer is a layer of longitudinal smooth muscle, which is part of muscularis externa. And then there's another layer of smooth muscle, which is circular smooth muscle. So that's smooth muscle going round in this sort of direction, so all the fibres are aligned in this direction, whereas the fibres that are in this section are aligned coming out of the page. So parallel with the direction of the lumen of the intestine. And then we have the submucosal layer, which ends with what I've drawn here is going to be muscularis mucosae. And then you have intestinal villi coming out like this. Okay, now draw next one of these villi zoomed up. So one of the villi, because obviously the bacterium is going to colonize the mucosal surface, so it's going to colonize the surface of these villi. So one of these villi might look like this, and it is lined by a layer of epithelial cells which are all cuboidal, and some of these will be filled up with mucus which they're secreting. So you'll see huge big white bulges which are the mucus. So here's mucus. And that's basically the structure of an intestinal villus, and then below it you'll have, well, go on and will form another villus, but below that then you'll have the muscularis mucosae. And then below that you'll have the submucosa, which will have loads and loads of submucosal glands, which are again lined by a glandular epithelium similar to this, and they secrete mucus. So the mucus is there to well, to protect the epithelium, to provide lubrication for the movement of contents and food boluses, and also potentially to protect the epithelium from the horrible enzymes that the pancreas and the liver are going to release into the intestine. Well, the liver isn't going to release enzymes, but it's going to release bile, and bile's not particularly nice, it's full of acid. Okay, so what are we going to do next? I'm just wondering about what I've just said. Bile's full of acid. You do call them bile acids, but I thought the point of bile was to neutralise the... Oh no, it's not. No, bile's purpose is to emulsify fat. I think there's some other way that they how does the oh, there's a physiological question. How does the how do you neutralize the stomach acid? I think it's secretions from. So that might be what these are. These potentially are secreting fluid to neutralize the acid as well from the stomach, especially if it's in the layer of the duodenum. Anyway, onwards with cholera. So cholera, our little bacterium is going here, and it's going to colonize, so it's going to divide many times, and you're going to get lots of Vibrio cholerae on the surface of your small intestine. So the next step is that it goes into your small intestine and colonizes the mucosal surface, and colonization then occurs. Okay, so now what's it going to do? It's going to start producing the cholera toxin. And the structure of cholera toxin is that it has one A subunit, and then it has 5 B subunits which surround the A subunit. So I'm drawing 3, but there'll be another 2 on the other side. So there's that one, and there's the start of that one. So you have like a pentagon of B subunits below, and then sitting on top of that in the middle you have an A subunit. So that's cool. So it's often referred to as an AB exotoxin because it's made up of an A subunit and a B subunit. In this case 5 B subunits, so AB5 it's often called. And this is cholera toxin. toxin, cholera toxin. Okay, so this bacterium is going to produce cholera toxin, it's going to secrete it, and it's going to go over to these epithelial cells. And these epithelial cells have in their membrane something called the GM1. Can you see this? No, you can't. So let me just bring this over here. The epithelial cells, in their membranes, they have something called the GM1 Ganglioside. Now, this basically it's similar to a phospholipid. It's got a different structure to a phospholipid, but it's basically got two fatty structures. I think this first one is generally arachidic acid, and then the second one isn't an acid. It's a long hydrocarbon chain as well, and it's not bound to glycerol. It's bound instead to 2-amino 1,3 propane 1,3 diol rather. So you have that structure I'll draw the structure of that. So this is propane. This is 2-amino. So you'll have an amino group here, and you'll have another hydroxyl group here. So you've got an amino group in the 2 position, and you've got 2 hydroxyl groups on the 2 ends, and you can't see this. Right, there we go. So this is the structure that we're going to use to bind these 2 together. So the acid forms an ester with here. So arachidic acid is a 20-carbon carboxylic acid. It's also sometimes called icosanoic acid, which makes sense because, you know, an icosahedron is a 20-sided shape. It basically is going to form an amide link with this amino group. So one of these hydrogens will come off, the hydroxyl group of this will come off, and they'll form a link basically between the carbon and the nitrogen. And on here, you might think that this is going to bind to the hydroxyl, but no, actually one of these hydrogens comes off, and this, I think it's a 17 carbon structure with 1 double bond between the first and second carbon. So over here you have a double bond, the first ever bond you have, and then this first carbon, basically it will be bound here. So you have a carbon, then you have a double bond, and then you have 15 more carbon atoms that are just linked basically by simple alkane linkage. And you'll have 2 hydrogens there, and 2 more hydrogens will come off each of these, and then off the final one there'll be 3 hydrogens, you know. And then off this you have a big complicated structure. So these two acids are going to sit in the membrane, and then this bit is going to sit outside the membrane, and there's a huge, great complicated structure attached here. It's loads and loads of glucose molecules and glucosamine molecules attached to each other. And you can Google it if you want to see a picture. So basically, our epithelial cells have this molecule, which, like that, GM1 ganglioside, in their phospholipid bilayers. And what's going to happen is that the cholera toxin, the 5B subunits of the cholera toxin, so I'll draw it again. So it looks kind of like this, and then there's 2 that you can't see. And this is going to come along, and here's this big complicated bit of, I know I've drawn it a bit badly, this suggests it's glycerol, but here's the phospholipid bilayer here. This is sitting in the phospholipid bilayer. This is going to come and bind here. And what it's basically going to trigger is it's going to trigger receptor-mediated endocytosis of the cholera toxin. So this is cholera toxin. So clathrin is going to come along, you're going to pinch the vesicle off, so you're going to get a vesicle in here with the cholera toxin bound here. And this is going to take it, it's going to go to the endoplasmic reticulum, so I'll draw a lamellae of the endoplasmic reticulum. It's going to fuse with the endoplasmic reticulum, and then you're going to end up with cholera toxin in the endoplasmic reticulum. And from here what happens is that this A subunit breaks off and goes into the cytoplasm. So we've now got the A subunit of cholera toxin in the cytoplasm. And it's going to go, and it's going to basically grab a molecule of NAD, nicotinamide adenine dinucleotide. Shall I draw out the structure of this? Can I be asked? It's okay. Yes, we'll do the structure of NAD. I'll get another piece of paper because it does help in understanding what's about to happen. So right, so you start with a ribose up here. This is quite a big molecule, I'll warn you now. So you start with a ribose group. So you start with ADP basically, nicotinamide adenine dinucleotide. So ADP looks like this. You have a carbon up there, you have a carbon here, you have a nitrogen here, a carbon here, a nitrogen here, you have a double bond there, a double bond there, you have to have a hydrogen there, you have coming off here a nitrogen, a carbon, up to a nitrogen, oh no, not a double bond there, just a single bond, so make that just a single bond, one single bond, then a double bond here, yes, and a single bond there, and off this carbon you have an amino group, and off this carbon you have a hydrogen, and that's adenine there, this bit, it's linked to ribose, this is an oxygen here, and up here you have a carbon, an oxygen, a hydrogen, a hydrogen. This oxygen is linked to a phosphate group, as so, which is then linked to another phosphate group. What have I done there? Which is then linked to another one of these basically. So off to a carbon, hydrogen, hydrogen, carbon, oxygen, carbon, 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 oxygen. And then instead of being linked to a normal organic base like adenine, it's instead linked to nicotinamide. And the structure of nicotinamide, I'll draw the structure of nicotinamide out. So you have a nitrogen here, and it's double bonded to a carbon. So this is similar to a benzene ring, but with one of the carbons replaced by a nitrogen. What's the fancy name for this sort of a ring? I think it's called a pyridine ring. Pyrimidine ring means that you've got two nitrogens. So you've got a benzene ring, but you've got two nitrogens replacing it, and the two nitrogens have to have a single carbon between them. So a nitrogen and a nitrogen there is not a pyrimidine ring, it's something else, I don't know what it's called. It's a nitrogen there and a nitrogen there, and that's a pyrimidine ring. And when you bind it with this sort of ring here, which is called an imidazole ring, you get what's called a purine, which is why adenine and guanine, which has a very similar structure to adenine, are both called purines. The other two organic bases, cytosine and thymine, just have this structure, they don't have this added on, and that's why they're called pyrimidines. But this is a pyridine rather than a pyrimidine, and you have up here, what do you have? It's an nicotinamide, so you've got an amide. And you have a hydrogen off here, you have a hydrogen off here, so it's just like benzene basically. So that's nicotinamide, that group here, and this nitrogen is obviously going to have a positive charge because it's got, oh wait, is it? Wait, you've got this, yes, because its lone pair is going to be donated to this, so it's going to lose electrons. Okay, so then you've got a hydroxide off here, so notice that these are not, you've got the same up here, these are not deoxyribose, these are ribose. So they're the sort that would be in RNA rather than DNA. And you'll have a hydrogen there, I think. I'm questioning this. Yeah, I think that's right. Hydrogen here and a hydroxyl off here. So yes, if this was DNA, this would be gone. It's in DNA you have 2-deoxy, and this is the second carbon of ribose. 2-deoxyribose in DNA. Okay, so that's the structure of oh damn, you can't see the whole thing. That there is the structure of nicotinamide adenine dinucleotide. And basically, cholera toxin A subunit is going to come over here, it's going to wrench this bit off. So the whole nicotinamide is going to come off. And what you're left with is what's known as an ADP ribosyl group. And obviously that's not a particularly stable bond. It's not a very fair bond. It's not a, it's a lone pair binding to this. It doesn't seem that like a normal bond. So this just wrenches off. you take this, and what nicotinamide adenine dinucleotide does with this ADP ribosyl group is it sticks it onto the intrinsic GTPase of GS protein. Okay, so a bit of hardcore biochemistry now. This pathway should be so familiar to you that yeah, you should never forget this. So you have I'm not going to draw the beta gamma subunit, and I'm not going to draw like a G protein coupled receptor. Here's our alpha subunit, alpha S, of the SG protein. And here we'll have the intrinsic GTPase. This is not a literal diagram. This is a what's the word? It's a picture. It's a picture. It's not a diagram. This is not what this protein looks like. It's a pictorial representation to make sense of this. And off here you bind GTP, and that activates the G protein. So this then goes off and activates adenylyl cyclase. It actually binds to adenylyl cyclase and makes the enzyme active. And basically, after a while, this enzyme is going to cleave the gamma phosphate off GTP, turning it to GDP, and that will inactivate this, and it will cleave off adenylyl cyclase, and the signal will be over effectively. But if cholera toxin adds ADP ribosyl onto this, it means that this no longer performs that role. So what happens is that GTP remains bound to alpha S, so you get complete overstimulation of adenylyl cyclase. So adenylyl cyclase makes cyclic AMP too much, so cyclic AMP goes up within your epithelial cells, and cyclic AMP activates the cystic fibrosis transmembrane regulator, which has actually a very similar structure to adenylyl cyclase. And the cystic fibrosis transmembrane regulator, obviously its name suggests that it's very, very important in cystic fibrosis, and it is, but it's also got a very important role physiologically as well, which is why cystic fibrosis is such an awful disease, because it's a mutation in this transporter which is very important. So this transporter, once it's activated, starts chucking chloride anions out onto the surface of the epithelium of the small intestine. So you start basically throwing away your chloride. Now if you throw away chloride, you can't just throw away chloride, it's got a negative charge. Are you just going to make your lumen a negative charge? You're going to make a capacitor? No, you're not. The sodium ions that remain in the cell will be attracted by the electrostatic forces, so the negative charge will attract the sodium across. So sodium will go out, so you'll get loads of salt on the outer surface of the epithelial cells, i.e. in the lumen of your small intestine. Now if you move salt, what happens? Osmosis. You make the little fluid that's out there much more concentrated, hypertonic. So then water moves following the salt. So it moves from an area of high water potential to an area of low water potential. So water starts moving into the lumen of the intestine, and when that happens you build up a huge amount of fluid in the small intestine, and that causes diarrhea, and it causes a rice water diarrhea. It's often said. It's said that when the diarrhea, if you put the diarrhea in like a bucket, it's supposed to look like the water that you get if you cook rice and then you drain the rice. That water that you get off is what diarrhea in the case of cholera is supposed to look like, which is very different from the sort of diarrhea you'll get if you get sort of Shigella or something, where you get blood. Basically your diarrhea is blood. That's called dysentery. It's so this is it's very distinctive. There are other types of diarrhea that will look similar to this, caused by different bacterium, that act in the same way. So for instance, enterotoxinogenic Escherichia coli basically releases a toxin that does exactly the same thing as cholera toxin, called cholera-like toxin, sensibly. So I think that's pretty much everything. Oh, no, it's not. So why is this so bad? Let's discuss why is this so bad. If you have horrendous diarrhea, what happens? So if you excrete away all your blood, all your body fluids, what happens? Blood volume goes down. If blood volume goes down, what happens? you get cardiogenic, well, not cardiogenic, you get cardiovascular collapse, basically. If your blood volume goes too low, you're not going to have enough blood to be able to meet the needs of the body, so you're not going to be able to pump enough blood around your circulation to actually supply enough oxygen to your tissues. So you're going to get what's the terrifying sounding syndrome called shock, which basically is the fancy term for what I've just described, that the cardiovascular system is failing, that it's not meeting the needs of the body, it's not delivering enough oxygen and enough glucose to keep the tissues of the body alive. And when that happens, you're going to get multi-organ failure, and it's a question of which vital organ goes first, and it's probably going to be the brain. So that's why it's so bad that your this bacterium has invaded you, because it's going to cause you to basically defecate out all your body fluids. It's going to dry you out, and then you're going to get cardiovascular failure and you'll die. But there is a very, very effective treatment for this, which is called oral rehydration therapy, which is basically just aimed at replacing all the electrolytes you lose and all the water you lose, which is very simple. So I think that concludes the cholera video. I hope you enjoyed it. It'll probably take me absolutely ages to upload, so thank you very much for joining me.